Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're going to continue our lesson today talking about protein synthesis. Okay, sometimes this is called the central dogma of biology. Today we're really going to be comparing DNA and RNA. Okay, so thank you so much for being here today, and we'll go ahead and hop into it. So, here are some similarities in between DNA and RNA. Let me go ahead and give you the full name of them, and then that'll make some of these similarities more clear. So DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Okay, so both of these, DNA and RNA, are both genetic material. They're used in different ways, but they're both used to express your genes. Okay. Both DNA and RNA, they are both nucleic acids, and we heard that in the name. That's what the Na stands for in both of these, is nucleic acids. Because they're both nucleic acids, they're both made up of nucleotides. Okay, nucleotides are the monomers for nucleic acids. As a reminder, nucleotides are made up of a sugar, a phosphate, and a nitrogen base. Okay, DNA and RNA, they are both used in different ways to make proteins. And over the next couple lessons, we'll really show how they're used and what those different ways are. Okay, both DNA and RNA have a sugar phosphate backbone. Okay, and I'll show you a picture that will make that more clear in just a second. We learned last time that DNA has a sugar phosphate backbone. RNA is the same way as well. And then in both of these, DNA and RNA, they both have the nitrogen bases in the middle. Okay, and three of those nitrogen bases are the same. DNA and RNA, they both have guanine, cytosine, and adenine, but RNA has a different nitrogen base other than thymine, which we'll look at here in the next slide. So those are similarities, okay? And try to remember these similarities and differences, because towards the end of this lesson, what we're going to do is look at a Venn diagram. So you're going to want to try to remember how they're alike and how they're different. So these are all the ways that they are alike. Let's look at some ways that they are different from one another, okay? So one major difference in between DNA and RNA is that the sugar is different in both of them. You might remember that they both have that sugar phosphate backbone. Well, the sugar that they are using is different. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. That's where those first letters are coming from. The deoxyribose stands for the D, and the R stands for ribose in the RNA. Okay. Another way that they are different, DNA is double-stranded and RNA is only single-stranded. Okay, so RNA doesn't really have that twisted ladder appearance because it only has one side of it. Okay, the way I remember this is that double and DNA both have that letter D in there. So that might be an easy way to remember that DNA is the one that's double-stranded and RNA is only single-stranded. What are some other ways that these two are different from one another? Well, DNA is contained in the nucleus, and for the most part, that's the only place it's going to be. It's safe in the nucleus, and we want to keep it safe, so we lock it up in the nucleus and we don't let it leave. The RNA, on the other hand, you can find it in the nucleus, but you can also find it in the cytoplasm. Okay, so that would be another way that they're different from each other. Some other differences here, RNA has the nitrogen base uracil instead of thymine. And that means in RNA, adenine binds with uracil. Okay, so look at that word, thymine. Thymine is only in DNA. RNA does not have thymine. Instead, it has a base called uracil. Okay, so try to remember those differences there. RNA is involved in making proteins, and DNA holds the instructions for making proteins. So they're both used to express your genes, but they're used in different ways. Okay? Great, so let's look at this picture really quickly. Yes, this is a picture that shows the difference in between DNA and RNA. Okay, on the left we have DNA, and you can see how it's double-stranded, right? It's got those two yellow lines that kind of curve around each other. Okay, on the right, that's RNA, and you can see that it only has one strand. It is not double-stranded like DNA is. Some other differences here on the left, you can see that the purple stands for thymine. Well, we wouldn't find that base in the RNA. If you look really closely, you'll see a different letter. You'll see U's popping up in some of those places. And so RNA has uracil, where DNA has thymine. Okay, so this is how they do kind of look different from each other. And we'll talk about some other differences in between RNA and DNA as we keep this going. Okay, so one thing I need you to know about RNA is that there are three types of RNA, and they all have different jobs. Okay, so one thing you'll hear me say a lot over the next couple lessons is mRNA. Okay, what does that stand for? Well, mRNA is a type of RNA, and the M stands for messenger. Okay, we're going to use mRNA to write a message. Okay, another type of RNA is called rRNA, and what that first little r stands for is ribosomal. Okay, ribosomes are made up of RNA, and so the type of RNA that they're made up of is called rRNA. And then the last type of RNA is called tRNA, and that T stands for transfer. Its job is that it's transferring amino acids to the growing polypeptide chain. Okay, so RNA plays an important role in protein synthesis, which has two important phases. The first part of protein synthesis is called transcription. Okay, when we transcribe DNA, what we're doing is we're making a copy of it, but that copy is in the form of messenger RNA. So remember, the DNA is in the nucleus. It has to stay there where it's safe. But everything we need to make proteins is out in the cytoplasm. Well, if you had to get a message, if you had to get information to somebody and you weren't physically around them, what would you do? You would send them a message, right? That's kind of what mRNA is used for. It's a way to transmit information across from the nucleus into the cytoplasm. Okay? The last part of protein synthesis is called translation. What we do here is we take that mRNA molecule that we made and we use it as a template to make proteins. Okay, so protein synthesis, it has two phases, transcription and translation. Transcription comes first. It's writing the code down, and it happens in the nucleus. And then once we have that message written down, we go out to the cytoplasm. We'll start translation, and that's actually making the proteins. Okay, so there are a couple of different types of RNA, and there are the names of them. You have messenger RNA, transfer RNA, and ribosomal RNA. Okay, so let's look at transcription in a little bit more detail, and we'll actually go ahead and get a little practice here on what's going on. Okay, so when we want to make proteins, a series of steps have to be followed, okay, in a very specific way. So first, DNA must remain in the nucleus. Remember, DNA is in the nucleus where it's safe. We have to keep it there, okay? But everything we need to make proteins is out in the cytoplasm. So how can we get the information from the nucleus to the cytoplasm? We're going to write a message, right? And that message is in the form of mRNA, messenger RNA. So what we're going to do is that ribosome comes into the nucleus, it reads the DNA, and it's going to make a copy of it, okay? That mRNA is almost exactly the same. We want it as close to the DNA as possible. Then that messenger RNA is going to leave the nucleus, it's going to go out to the cytoplasm, it's going to find a ribosome, and then translation is going to start. And translation is how we're actually building the proteins. Okay, so what I want to do here is we're going to do a little bit of practice. Okay, so what I need you to remember here is that in RNA, we don't have the nitrogen base thymine. Instead, we have a nitrogen base called uracil. And uracil pairs with adenine. So what I'm going to do, I'm going to give you a DNA sequence, and I want you to transcribe it into messenger RNA. And you're going to use your complementary base pairs rules for this. So just a quick overview of that. Cytosine always pairs with guanine, and anytime you see an adenine in the DNA, we're going to pair it with uracil. And then sometimes you'll see a thymine in DNA, and that's going to pair with adenine in the messenger RNA. So I'm going to put the DNA sequence up here. I want you to take a second and see if you can figure out what the mRNA sequence would be. Okay, so let's look at this real quick. Here's the DNA sequence. It's GGAACCTTA. Take a second and try to remember those base pairing rules and see if you can come up with what the messenger RNA is going to look like. Okay, I'll give you a couple seconds to do that. Okay, so that's the DNA sequence. If we were to transcribe it into messenger RNA, this is what it would look like. Okay, so notice how these base pairs are pairing up here, okay? Anytime we see a G in the messenger RNA, there's going to be a C because those two bases are complementary, okay? Anytime you see an A in the DNA, we're going to use U in the RNA because they're complementary and RNA does not have the thymine, okay? And then I see a couple of C's. Well, those are going to pair up with G's in the messenger RNA, and then the TTA is going to turn into AAU, okay? So we kind of talked about the complementary base pairs for DNA, but you also have to be able to transcribe it into mRNA. And the main rule there is that you don't have thymine in RNA, you use the base uracil instead. Great. Okay, so let's keep it going. So that's a little overview of transcription. Let's talk about translation next, okay? So translation, this is happening in the cytoplasm. What's going on here is that messenger RNA, we made that in the nucleus. It leaves the nucleus. It comes out to the cytoplasm, okay? When it gets out to the cytoplasm, it's going to find a ribosome. Remember that ribosomes are made up of rRNA, okay? And it's going to attach to that ribosome, okay? It's going to read the mRNA in 3 bases at a time, okay? We call that a codon. So a codon is 3 nitrogen bases on an mRNA sequence. What does a codon do? Well, it codes for an amino acid, okay? So it codes for an amino acid. This is how we're going to build our proteins. Remember that proteins are made out of amino acids, okay? So the ribosome is moving down the mRNA. It's reading it in sets of 3, okay? Once it finds a codon, once it can read one of those 3 nitrogen bases on the mRNA, it's going to find a tRNA that is complementary with it. On the tRNA molecules, there's 2 things you kind of need to know. It has something called an anticodon, which is 3 nitrogen bases that are complementary to the mRNA, and it also carries or transfers an amino acid. Okay, so what we're doing here is the tRNA and the messenger RNA are going to find a complementary base pair. When they pair up, the tRNA leaves the amino acid for the ribosome, and the ribosome is going to start building that protein. At this point, we are really going to call it a polypeptide. And the reason for that is the base in between each amino acid is called a peptide bond. So if you get a lot of peptide bonds, it's called a polypeptide. Okay? And then the tRNA is going to carry those amino acids, so we're building our polypeptide amino acid by amino acid. I know this stuff is really confusing. We're going to look at some pictures that kind of clear this up because it's all very hard to conceptualize. So let's take a look at transcription. This is basically a picture of transcription. What's going on here? Well, first of all, we are in the nucleus, okay? We need to know transcription happens first. It happens in the nucleus. What's going on is you can see that this is DNA. And down there at the bottom in the middle, that DNA molecule has been split. And we kind of talked about how that happened last time. We use an enzyme called helicase to basically unzip this DNA. Okay, well what happens then is we're going to make our mRNA. Okay, we're going to make a copy of the DNA in the form of RNA. And that copy is called mRNA. We're using an enzyme called RNA polymerase, and what it does is it goes down and reads that DNA sequence, and we're pairing up complementary base pairs. Notice how on that RNA transcript we don't see any T's. We see U's instead, and that's because RNA does not have thymine, it has uracil. So this is going to continue until the whole gene is copied down. Once the gene is copied, that mRNA molecule is going to leave the nucleus and move out into the cytoplasm where our next step is going to happen. This next step is called translation. Okay, I know there's a lot going on here. Please don't panic. We're going to look at transcription and translation in a lot of detail over the next 3 or 4 lessons. I just kind of wanted to introduce it. So don't panic. You're going to see this picture a lot, and we're going to talk through it a lot over the next couple of days. Okay, so what's going on here? Well, this is our mRNA molecule. We just made this in the nucleus. Okay, so now it's leaving the nucleus, and now we're in the cytoplasm. This is that copy of the DNA, okay? It's going to meet up with a ribosome. The green here is a ribosome. Ribosomes have a large subunit and a small subunit, and they come together to form a full ribosome. Okay, well what happens then is this mRNA starts to move through the ribosome, okay? And the ribosome is reading this mRNA 3 bases at a time. A 3-base set is called a codon. So I know it's kind of hard to see, but it's reading it 3 bases at a time. That's called a codon. Then what happens, these little things floating around out here, these are tRNA, okay? And what happens is they have, at the bottom, they have what are known as anticodons. When an anticodon and a codon are complementary, it's going to come into the ribosome, and those two, the anticodon and the codon, are going to connect. When that happens, the tRNA is going to drop off its amino acid. Each of these little circles here is an amino acid, okay? And then once it's dropped off its amino acid, the ribosome makes a peptide bond in between the amino acids and links them together. Then the tRNA leaves, the mRNA moves down 3 more bases, the RNA is going to read the next codon, and the whole process starts over. Okay, so this is how DNA and RNA pair together to express your genes. Okay, this is why it's known as the central dogma, is because whatever is in that DNA, whatever code is in that DNA, that's the proteins that your cells make, and your body's mostly made up of protein. So basically, your DNA is controlling what your body does through this whole process of transcription and translation. Okay, so that's an overview of protein synthesis. Like I said, we're going to look at these in a lot more detail over the next couple of units. What I wanted to do now is I have a Venn diagram that compares and contrasts DNA and RNA. So what I'd like for you to do, teacher, if you could go ahead and pass out that Venn diagram. Okay, why don't y'all take 5 or 10 minutes to work on it, and then when you're ready for a review, I'll do it on the board up here, and that way we can kind of walk through it together. Okay, so take a couple minutes to do that. I'll get it pulled up, and here in just a couple of minutes, we can look at this together. All right, hopefully this didn't give y'all any problem, but just in case it did, why don't y'all just follow along with me. Feel free to change or add anything you don't have, and then that way this is going to be a great resource to use to study when you're trying to get ready for this test or when you need to look over it, okay? Because what we're going to do is we're going to put all the information that applies to only DNA over here, anything that applies only to RNA here, and any similarities they have is going to go in the middle. So this is going to be a great resource to review. So let's go ahead and hop into it. Okay, the first one I want to do is genetic material. Which of these two is genetic material? Well, if you remember from the lesson that we just went over, they are both forms of genetic material. Okay, so that goes in the middle. They're both genetic material. Okay, the next one says deoxyribose sugar. Which of these two has deoxyribose sugar? Well, that's the DNA, right? That's what that D stands for. So let's write deoxyribose over here. Great. All right, the next one says in the nucleus. Which of these two is in the nucleus? You might want to say DNA, and you're right, but remember that we can also find RNA in the nucleus. So this one actually goes in the middle. Okay, in the nucleus. All right, the next one says thymine. Where should we write thymine here? Only DNA has thymine, so we're going to write that one over here. Great. All right, going up to the second column, this one says ribose sugar. Well, where should we put ribose sugar? Well, that's the RNA, right? And I'll go ahead and write sugar over here as well. Okay? So that's what these letters are standing for. Deoxyribose is where we're getting the D, and the ribose sugar is where we're getting the R. The next one says in the cytoplasm. Okay, well, we can only find RNA in the cytoplasm. Okay, DNA is in the nucleus where it's safe. Okay, double-stranded. Which of these is double-stranded? That's the DNA, right? They both have the letter D in there. That's a good way to remember that. So this one is double-stranded. Great. All right, the next one says single-stranded. Well, if DNA is double-stranded, the RNA must be the single-stranded one, and you would be correct. Okay, great. All right, that third column, this one says nucleic acids. Well, nucleic acids is going to go in the middle. And that's why they're DNA and RNA, right? That stands for nucleic acid. So just looking at the name should kind of be a hint on that one. All right, the next one says made of nucleotides. Well, because both of these are nucleic acids, they're both going to be made of nucleotides. Okay? Great. All right, the next one says adenine. Where can you find adenine here? Well, that actually goes in both of them as well. They both have adenine. All right, the next one says has 3 different types. Which of these two, DNA or RNA, have we talked about having multiple different types? Well, the RNA has 3 types. And you might remember that that's the mRNA, the rRNA, and the tRNA. RNA has 3 different types. Okay? The next one here says sugar phosphate backbone. Okay? Well, that actually goes in the middle. They both have a sugar phosphate backbone. Okay? Can't quite fit it in the middle, but that goes in the middle there. Okay? The next one says used in transcription. Which one of these is going to be used in transcription? Okay? Well, they're actually both going to be used in transcription. I'll write it over here. Transcription goes in the middle. Great. All right, the next one says nitrogen bases in the middle. That also goes in the middle. So I'm going to abbreviate this. Nitrogen bases, NB, in the middle. Okay, a lot of similarities in between these 3. All right, the next one says guanine. Where can we find guanine? Guanine is in both of them. All right. The next one says cytosine. Can you find cytosine in DNA? Can you find cytosine in RNA? You can find them in both. So cytosine also goes in the middle. All right, the next one says uracil. All right, this one does not go in the middle. Which of these two has uracil in it? That is the RNA. So we're going to put uracil over here. All right, great. This next one says used in translation. Okay, well, we put that transcription is in the middle, that we use DNA and RNA in transcription. But if you think back to it, DNA is not actually directly used in translation. So we're going to put translation over here in RNA. And as we're going over transcription and translation in more detail over the next couple of units, that's going to become more clear. But for now, let's just write translation over here on the right. All right, the last one here says has hydrogen bonds holding it together. Okay? Well, the hydrogen bonds is really only in the DNA because RNA is single-stranded. It doesn't have to hold those 2 strands together. So we actually have the hydrogen bonds is going to go over here. All right, great. Y'all, thank you so much for following along. I hope that kind of made it more clear. Your teacher has some more assignments for you to work on, so make sure to give that your best effort, and I hope everybody has a great day.